Yeah, thanks, and thanks for the invite. We are in Metabolomics Australia. We always like to come and showcase what are the capabilities we have in our facility, and then we make researchers to think how they could use our facility. So today I will be showcasing the spatial metabolomics platform, and then I will just give a quick snapshot of what are the capabilities so we have in the next 10 to 15 minutes. So we are Metabolomics Australia. So Metabolomics Australia is funded by its NCRIS, and we are part of Bioplatforms Australia. We have different nodes in different states, and so depending on the expertise, so the nodes are specialized in those specific areas. And we are in Bio 21 here, which is the next building right to WEHI, and then we are specialized in various field, so whether it could be biomedical, plant, environmental, and also the imaging. And we are the only one who have imaging within all our nodes. So we have plenty of instruments. We are located in Bio 21. As I said, we have plenty of instruments for various analysis, whether it could be GCMS, LCMS, and also imaging system. And we like to analyze, or we have done work on various sample types. So here are some of them which I have mentioned briefly. And then we analyze thousands of metabolites, and we are really good in that across various metabolomics pathways, across various chemistries of the metabolites. So any questions, you guys please come and have a chat with us, and then we will be able to help you guys in that space. So today, as I said, I will be mainly focusing on the MALDI, which is the spatial metabolomics area. So what is this MALDI mass spectrometry? As the name suggests, so the MALDI mass spectrometer is it's basically here, so you will be having your section of interest, so whether it could be a sample or a tissue section, and then you spray carefully chosen matrix on it, whether it's a sublimation or spraying, and then these matrix will dissolve and then pass on the proton from the matrix to your endogenous metabolite, and these metabolites get into the mass analyzer. And then so what do we do with those data? So just the masses, what we see from the mass spectrometer, either if you are looking for the Dunkin' Donuts in US, where you should see a spatially resolved where it is present, or whether if you're looking in your tissue section a specific chemical map or a chemical signature where exactly it's coming from the region of interest in a specific region, you can spatially map them out from our MALDI mass spectrometry imaging technique. So we used to have MALDI FTMS before in our institute, which was getting quite old, and we had to transfer that to the current technologies. So it's upgraded that to the IMS. It's IMScope, which is the MALDI mass spectrometry technology platform we have. And we transformed the technology from FT-ICR to TOF for various reasons, whether it could be it's easy to maintain flexibility, or but we were mainly looking for the high spatial resolution, and also we wanted to have a faster throughput so that more researchers can make use of the facility. And then we wanted to also have a single cell analysis capability also, and we wanted to increase the sensitivity to whatever we had before to a better sensitivity so that we can get a better coverage of the metabolites and also reproducibility. And we also wanted to have an economical in the ongoing cost so that more researchers can come and use the facility. And then the workflow has to be it's user friendly, so that whether it could be a postdoc or a master student, so they should be able to come and get trained and then so use the platform as soon as possible without much complexity. And also, it also fits our subscription model, to which I will take you through later. So flexibility, when I mean flexibility, so basically the instrument what we have upgraded now, which is capable of both, whether it could be an MALDI imaging system, or you can do a complementary analysis where you can put an LC next to it and then you can do the LCMS, or you can do the MALDI imaging system within the same instrument. And this instrument is also embedded with the microscope, which is in the right front of the system, so that helps us to take the optical images of the tissue sections before collecting the MALDI data. As this is a TOF technology, we are very surprised to see that it has a high mass accuracy, even though we don't calibrate with external chemicals, so that it can suppress our signals too. So we don't really need to calibrate for up to 2 weeks too, because it's really holding the mass accuracy for less than 2 ppm. How it is important? Having an high mass accuracy for a long time, so basically it can collect the data, so basically in high mass accuracy, so that it will also allow us to precisely identify not only the lipids, with less than 2 ppm mass accuracy error, but it has also allowed us to explore in the low molecule space to where we can screen the central carbon metabolites with the masses which are less than 400 or 500 MOSEs. So how does the microscope helps us in terms of the MALDI platform compared to what we had before? So it helps us to take the images in various resolution, the optical resolutions here. So this happens before actually we collect the data. Once we collect the mass spec data, it helps us to co-localize the microscopic images along with the MALDI images so that it can show us the precise co-localization. So in this case, we are we have a variability of less than 5 microns. This is really important, which has never been seen before. So how does this co-localization variability that is less than 5 micron helps us? It helps us to tap onto the single cell metabolomic space where here now, so you can culture your cells on a specific slide, and then we can even get the cell specific signatures of metabolites from a single cell platform too, as an example. So here we have done the spatial resolution in this instrument can go up to 5 microns, which is really amazing compared to the one what we had before, which can go up to 20 or 25 microns. Now we can tap onto up to 5 microns. So that helps us to go to a single cell specific up to 5 microns here. So here you can see the segmentation analysis of the single cell specific and also the cell specific signatures of your metabolites too. So to get all these pretty images, we need to have a good sample preparation workflow. So for that one, we have made it quite easy in terms of so that for the researchers who are expert in doing the sections, they will prepare their own sections. But for the researchers who doesn't know how to do the sections, we have the Melbourne Histology platform here. They look after doing all the sections for us. And once the sections is done, as I mentioned previously, we need to carefully select the matrix and the type of technology for it to be deposited onto the matrix, it's onto your tissue section. So that can be done either with the sublimation or with the spraying protocols. And we have also streamlined in this space here. So these are very easy to use, and even for the students, master students, they can easily come and learn this within half an hour, and they can be an expert in running these instruments as an example. So these instruments are really good in terms of controlling the humidity, and also you can also control how much thick this matrix can be deposited on your tissue sections, and also this is reproducible, so how many times which you wanted to use for your replicates. Depending on what type of the technology, what you use in terms of to deposit on your tissue sections, it is really important. So that depending on whether you are using the spraying method or whether you are using the deposition method or you are combining both together, the crystal size improves the sensitivity and also the spatial resolution. So we have seen some really good data when you when we combine both spraying and the vapor deposition. So we can go the spatial resolution up to 5 microns, and also we have improve the sensitivity of the small molecules that we haven't seen that before in this platform. Okay, so in terms of the work, preparing the instrument, the workflow here, it's quite straightforward, and it takes very minimal time in terms of preparing the instrument for you to collect the data. The data acquisition of a specific sample purely depends on what type of spatial resolution which you guys are interested in. So if you're really looking for really high spatial resolution data that is around 5 microns, for a normal mouse brain section, it takes around 13 hours for you to collect. Or if you're looking for around 40 microns, so basically then it only takes 2 hours for you to collect the data across the sections. So this model has allowed us to work in 2 different subscription models. In Metabolomics Australia, we don't have subscription model because our facility has to run 24/7 to work on various researchers projects. We can't really afford the breakdown of the instruments. So, but this kind of user-friendly has made us to it allow the subscription model. So what is the subscription model here? So basically we can train researchers like PhD students, postdocs, and they can come and use our facility, and they will be doing all the data collection, and also they will be analyzing the data by themselves so that they know exactly what happens behind the background, and the cost will be minimal in this model. Whereas in the non-subscription model, which is very common model in Metabolomics Australia, so basically, so we do everything, and then we just it's hand over the data. So the benefits of this is basically the researchers can focus on the other things and leaving these to the experts to do the job here. Okay, so here is a quick snapshot of when I really mean, so what can be achieved from this instrumentation is if you're really having a 10 by, if you want to monitor what's happening within a 10 by 10 micron section, basically we can do multiple experiments within that small 10 by 10 section, whether you can collect the full mass data or one mass data and with multiple MS. So what does that allow us? So basically, so that's your full scan data. And then, so if you want to see what's the composition of a specific mass, for example, in this case in PC384, which we have identified from the ion, we can also do an MS of that so that you can see what type of fatty acids that are present within that specific lipid. And then you can also extract the specific images, the spatially resolved images within the tissue section too. Okay, not only the normal metabolites, we can also trace the labeled metabolites. So here is an example of a mouse lesion which has been labeled with and without the DTO label, and you can also trace the labeled signatures within your tissue section too. Okay. As this is a tough technology, there is no much limitations in this space of collecting the data, whether you can collect the data by narrowing to a small molecule region, whether it could be a small molecules, or whether you can narrow it to the lipid molecules too, or you can collect the whole data with a little bit of compromise in the data, and then you can get the entire profile within its one screen. So to get all this beautiful data, there needs to be a reliable software, and it has to be user-friendly for all the researchers to use. So Shimadzu has thought a little bit over into it, and they have combined all the data analysis platform into one package, where they have combined all the statistical analysis and also the data extraction, and also in terms of the metabolite, its annotation, everything has been combined in one software, which is quite straightforward, and it's user-friendly. So here is a snapshot of the statistical analysis and also the segmentation, its analysis, and also the extracted ion images, its images of a specific its matrix, and also the metabolite, its annotation of the individual masses towards its metabolites. So in terms of the statistical analysis, the software also allows us to do the PCAs, so whether it could be within the tissue section or between the different tissue sections, so whether it could be the control versus the treated, it all can be done within the same software. So saying that not everything is beautiful, so there are some limitations to the software, where one of the limitations is basically we can't really take the data from the software to directly try to annotate the metabolites through the public databases. So our bioinformatics team in MA has come up with the Shiny Cardinal app, where you can take the list of the masses that's been shown from your software, and then you just put that to the public, so within this software here. Thank you. And then you should be able to use the HMDB or the public available databases to annotate your software. And then so this also allows us for various things. So the things which we couldn't see in our software, we have also implemented, like checking the ppm errors also. So what's the ppm error of the specific metabolite? Its annotation which you have identified. And also for the large and the multi-omics data, so which Jürgen, so this morning was presenting, so we are also collaborating with Saskia Group and also Sarah Group here to try to come up with the cardinal workflows where you can compare multi-omics data, like the metabolomics data, with the transcriptomics and also the genomics data, so that how you can combine them and then compare the data together. Okay. So saying that, it's installed this instrument nearly like a year ago, and we have been helping a lot of researchers in various research fields, whether it could be human, mouse, or it's environmental, and also in the single cell space across the various different methodologies what we are currently using now. So if there is any questions in terms of your research, please come and see us. So here is our lovely team in Metabolomics Australia and Bio21 here, and I have put some of the contacts here. Please do take that one, and then so just get in touch with me, and I'm more than happy to take your questions now. Thank you.
Thank you.